It's truly an honor to be here tonight and to honor Gobind here at the University of Wisconsin. I have so many wonderful memories of being here, and probably at the foremost is learning how to do good science in Gobind's lab. And you had to focus on a project, as Tom mentioned, intensely. And most important to me is Gobind worked at the edge of what you could do. He'd be out on the horizon when everybody else was in the morning sunlight. He'd be out there trying to figure out what to do next. And he had the moral integrity to make sure that everything he did was right. And he wouldn't publish until it was absolutely right. And then he would publish it. And so I learned a lot from Gobind in those early years of my career. And I also, while I was here in Wisconsin, I think I not only worked harder, but played harder than I ever did anywhere else in my life. I shared a house, a Frank Lloyd Wright designed house, with Peter Bessemer, who's in the audience, and Ian Molyneux. And I swear we must have had dinner parties every other night. And then we'd come back to the lab afterwards and work for 2 or 3 hours. So, and traveled all over the state and saw everything from Door County to the Mississippi Delta, and I really had a wonderful time, but also had an intense life experience learning how to do good science with Gobind. I'm at the University of Wisconsin, and this is where I live. Down in the valley here is Boulder, Colorado, and then these are the foothills, and the mountains just above Boulder. These particular mountains are called the Flatirons, and that's because they look like an iron on end. And in this picture, I wish I could take credit for it, but it was taken by the university staff, obviously shortly after a light snow and a moonlit night. As some of you who have known me for some time may have noticed, I've lost a few pounds recently. And this is where I put a lot of them, because I did a lot of walking here in the foothills during the course of losing that weight. Let me think. I think this should do it. There we go. So, wait a minute, I've skipped too many slides here. How do I do this? Let's go back. Yeah, there we are. Well, I'll only try to touch on a few things about Gobind and his work, try to fill in a few things around what Tom has already said so beautifully. Gobind started his work in the nucleic acid field, learning how to synthesize dinucleotides using dicyclohexylcarbodiamide as a reagent. And this is one of the first papers of a syntheses published in a series of papers in 57 and 58, where he used carbodiamides to activate phosphates to form internucleotide linkages. And this served to catapult Gobind's career into, with Marshall Nirenberg, solving the genetic code, using this chemistry to make repeating trinucleotides and the triplets of the genetic code. Now, Gobind was one of the few scientists in those days who really practiced what's now become very popular, and that's bioorganic chemistry. And this is taken from a Federation Proceedings Abstract 1960, where he writes, chemical structural studies, physical chemical studies, elucidation of the mode of action of enzymes of nucleic acid metabolism, and in short, understanding the biological functions of the nucleic acids in precise chemical terms will all be profoundly influenced by polynucleotides synthesis. In the distant future, the total chemical synthesis of macromolecules possessing biological function must also be considered. The problems are vastly more complex than anything previously undertaken by experimental organic chemistry. Nevertheless, biology urgently asks, will organic chemistry extend its horizon and accept the challenge? Of course, the answer was a resounding no. It took organic chemists till at least 1990 before they recognized that there was a field called bioorganic chemistry. And the only thing most organic chemists would do was to take a very complex natural product and develop a 20 to 30 step synthesis to try to make it, and then they wouldn't do anything with it. But now, of course, bioorganic chemistry is very popular in organic chemistry circles, but not at this time. Gobind was a pioneer in trying to use organic chemistry to solve biological problems. And this comes up over and over in his work. I got to get this direction straight now. Tom mentioned after the genetic code work, Gobind was very intent on developing methods for synthesizing genes. And this was the first gene, alanine tRNA, synthesized here at the University of Wisconsin, published in 1970 in Nature, using a combination of chemical and enzymatic synthesis. This particular schematic shows the synthesis of a 20-mer carried out by Hans Weber, which was this sequence right here, which is part of this gene. That took Hans Weber 2 years to synthesize. That was his postdoctoral project. And then we joined these together, and this shows the last step enzymatically. We use T4 ligase, where we join 2/3 of the molecule to 1/3 of the molecule to form the final product, as shown here. And as I mentioned, announced the synthesis in 1970. And we were quite happy with this. And so we published the in detail synthesis of this gene in the Journal of Molecular Biology as 13 papers, December 1972. Well, Nature kind of reviewed the paper, and the title of their review was the journal where we published these 13 papers. And their commentary, it's much longer than this, but it goes something like this. Starts out with "Is enough too much" as the title. The 13 articles from Khurana's group, which comprise a complete issue of the Journal of Molecular Biology, surely sets something of a record. Every step of the synthesis has obviously been executed with consummate skill, and the whole constitutes perhaps the greatest tour de force organic and biochemists have yet achieved. I can agree with that. It was a tour de force, about 70 or 80 man-years of work, but it was out on the horizon, trying to do something that only a man like Khurana could conceive and carry out. And then Nature's correspondent goes on to say, like NASA with its Apollo program, Khurana's group has shown it can be done, and both feats may well never be repeated. January 1973. Well, years before, Gobind had stated in 1968, and several other articles I could have cited, how in the years ahead, genes are going to be synthesized. That was 5 years before this was published. The next steps would be to learn to manipulate the information content of genes and to insert them into and delete them from the genetic system. When in the distant future all this comes to pass, the temptation to change our biology will be very strong. We're doing all that today, thanks to Gobind's early pioneering work. But you know, molecular biology had a penchant for not thinking that this field was at all very interesting. I remember in 1975, after I'd started at the University of Colorado, I was asked to review the nucleic acid synthesis area at a Gordon conference. So I did. And then that evening, sitting around a table having a beer, someone who's now a member of the National Academy, is a molecular biologist, said, "Marv, why in the world are you trying to learn how to synthesize DNA? I mean, sure, Khurana used it to help solve the genetic code, and now he synthesized the gene, but what else are you going to do with it? What else are you going to do with it?" And that was the standard comment and attitude of the day. What are you going to do with DNA chemistry? There's nothing really useful about it. But we persisted, and in the early 80s, late 70s, we published this chemistry. Mark Matucci, as a graduate student, joined my lab in 1976, and I suggested we try to learn how to synthesize DNA on a polymer support, and he developed this procedure here. Serge Bouchacage joined a couple years later and developed a synthon, which we call phosphoramidites, which turned out to be the method of choice for putting together synthetic DNA. And so the cycle goes something like this. We start with the nucleoside on the support, take off the dimethoxytrityl group, add the phosphoramidite activated by tetrazole to form a phosphite triester, acylate any unreacted hydroxyl groups, and then oxidize the triester to the P5 derivative, and then we go around the cycle again. And this cycle takes all of a couple minutes at most, maybe a half a minute if you push it. And the yields are essentially quantitative. These days, and then of course we put this in a machine and the rest is history. But these days, if you ask biologists who figured out how to synthesize DNA, and probably half of them would say, well, Federal Express. Because all they do is type in a sequence on a computer, hit the return, and the next day their DNA arrives by Federal Express. So that's kind of where it is these days. There are, however, areas where there's a lot of development. For example, I've had a lot of fun over the last few years working with Agilent on developing how to adapt this chemistry. And the cycle I just showed you is still the state of the art, even though we developed it in 1980, is how to adapt this chemistry to DNA synthesis on a chip. And I've been working mainly with groups out in Santa Clara headed by Emily LaProst and Doug Dellinger, Doug's a former student of mine who spends time both in Santa Clara and also in Boulder at the facility there. And so what we learned to do is to reduce this to 3 steps: coupling, oxidation, and deblocking. And we actually carry out this chemistry in, come on, let's hit it. We couple, and then we carry out the chemistry in 2 different modes. We do the coupling step of about 20,000 or so features on a chip using a modified inkjet printer to do this. And then we transfer the chip to a flow cell where we do the oxidation and deprotection. And one of the major problems we had to figure out was preventing depurination. You see, as these are the little features where we're synthesizing DNA on this chip, and they're less than 100 microns in size. As the acid treatment step flows away from those spots, they're hydrophilic and the surface around is hydrophobic. So you get a very high concentration of acid here as this solvent evaporates. That causes enormous amounts of depurination. So we had to revise this by reintroducing the oxidation step over here to flush the acid out of this step here. And we basically eliminated depurination. And that has allowed us to move forward to where we now can routinely synthesize oligonucleotides on a chip. And if that chip has 20,000 features, we can synthesize 20,000 or so, where we make 150 mers with an error rate of about 1 in 250 base pairs, with about 47% of the sequences perfect matches. And the yield in excess of 99.5%, no depurination. And this shows some of the results here. The interesting part of this slide is that you can, of course, imagine if you have a nested set of PCR primers, you could isolate all 20 or 30,000 oligonucleotides and then bring in your primers for a particular nested set, and you could synthesize a modified gene or part of a chromosome, and then bring in another set of nested primers and synthesize a modified one, and you could make all kinds of mutations just from what you made on one chip. So this is pretty much where it is today, and these kinds of claims are far in excess of anything you'll find in any other attempts to make DNA on a surface these days. So what else have we been doing? That project was in collaboration with Agilent, and it's been a lot of fun because there's no way I could imagine having the engineering capability of building a modified inkjet printer that could, with surfaces that could make DNA. But our expertise in DNA chemistry helped bring this together. So we've learned in recent times to synthesize several interesting analogs, one of which is shown on this slide here. It's borine phosphonate DNA, where we introduce borine at different sites, either completely borine phosphonate or mixtures of borine and phosphate. This work was mainly carried out by Adrian Higson as a postdoc to start with, and I don't have a picture of him, continued by Heather as a graduate student, and then more recently with Agnieszka and working with an undergraduate, Mary Noe, who's now out at UC San Diego in the chemistry department. And Mary, as an undergraduate, actually completed this project. The other students here work with phosphonoacetate DNA, which I can see from the time I'm not going to be able to talk about. So we learned how to synthesize borane phosphonate DNA using a synthon with a 5' silo group, 3' phosphoramidite, and a nucleoside on a polystyrene support. The trick we had to figure out, and this was really challenging chemistry, was to how to protect the bases. And we ended up using acid labile or carbamates that are non-reducible to block the bases. And after carrying out this synthesis, introducing either borane and then acid to take off the silo group, HF acid, hydrofluoric acid, or peroxide to oxidize, we could mix and match and make oligonucleotides of any particular combination of borane phosphonate and phosphates. And to our surprise, these compounds are taken up by cells in the absence of cationic lipids. We've been working for some time now, 3 or 4 years, on how to synthesize a phosphonoacetate linkage where various ester groups protect the oligonucleotide chain so that we can get these ester functionalities into cells in the absence of cationic lipid, and then have them come off and keep the DNA into cells. But now we've discovered that borane phosphonate oligonucleotides are taken up the same way. Let's see, we're running a little bit late here, so I'm not going to, I think what I'm going to do is skip a few things. This is just a control slide showing if you take phosphorothioate DNA and a FACS analysis, and you look at the background fluorescence. You see it here. You add thioate DNA with lipids. You see fluorescence marking the uptake of fluorescent oligonucleotide into cells. And then this is the one, phosphorothioates without cationic lipid. This is no news. These are controls. And then if you take the phosphonoacetate protected, where these are all phosphonoacetate linkages, any one of these three, and here's the phosphonoacetate itself, ionized in the center of the molecule. So you have 21 mers, of which 5 are charged and the rest are neutral. And these oligonucleotides are also taken up by cells. Remember, the background fluorescence is way down here, and so you get about a 1,000-fold increase in fluorescence in these oligonucleotides. And we show that these will also serve as carriers to take up interfering RNA. And you can see here that here's one of those oligonucleotide siRNA duplexes, which inhibits the production by, here's a Western blot, of the production of this lamin AC protein almost as well as the Tuschl assay. We're using thioates plus lipids as shown here. And then if you look at the amount of RNA produced when you use these carriers, phosphonoacetates to carry in the interfering RNA, you see that there's downregulation of mRNA almost to the same extent as phosphorothioate siRNA. We are now learning in some unpublished work with, in my lab, we've learned that here's the data that borane phosphonate DNA, this is a 21-mer at 12 nanomolar concentration. Here's 1 micromolar, several-fold higher. And here's a control background, no lipid, no DNA. And neither one of these samples had any lipid at all in it. And you can see the uptake of this fluorescently labeled borane phosphonate DNA. And when you increase the concentration, you see higher fluorescence. It's down here rather than here. So these compounds are obviously taken up in the absence of cationic lipids, so we're going to have a lot of fun seeing what we can do with those. Another interesting experiment is that we've discovered that this borane phosphonate linkage reduces metal ions. For example, here's a synthesis of a 16-mer and here's a 20-mer. The peaks are quite broad. This is all 16-mer and 20-mer, but remember these are all chiral centers, and so the peak is very broad. But if you spread this out on a gel and you get this band of borane phosphonate DNA, and then you expose it to silver nitrate overnight, you see an intense coloring of the borane phosphonate band. You don't see this with phosphate. So that tells us that the borane phosphonate linkage is reducing silver ion to silver metal at that site. And we can also do this with, here's a compound 12 long, 12, 12. Here's with lead bromide. You see another band reducing lead, and here's silver chloride. gold chloride. You see gold being reduced as well. And this is the position of normal DNA on the acrylamide gels. And you see there's no banding there at all. So we're specifically reducing metal ions. Well, to paraphrase what this fellow at the Gordon conference said, what are you going to do with it? Well, maybe we're going to build DNA wires. And we're going to make them 20 or 30 long. We're going to put all kinds of circuitry in there and maybe even some small transistors, and we'll see what we can do as time goes by. But maybe we can use this chemistry to make small DNA wires on a surface. Another project we've been pursuing is to build oligonucleotides with phosphorus metal linkages, and this is a series of paper of syntheses carried out by Andrei Pav in the lab. And the interesting aspect of this is it's going to be a very challenging synthesis. We've only made dimers. The problem here is that that linkage is labile to everything except acid and extremely neutral, non-nucleophilic reagents. So the whole scheme of synthesis has to be designed around those kinds of conditions, protecting groups and everything. And that's going to be a fun challenge to try to meet. So this is where we are on this. And you can sort of see what I'm talking about here, because here's thymidine without any protecting groups. You get the highest yield. All the others with protecting groups, you get much lower yields. And the reason thymine's lower is because you need base to take off the oligonucleotide from the support. So there is some degradation even with thymidine. But the yields are very high, but the chemistry of protecting groups is unimaginably going to be complex. And it's going to be fun to see what happens. So I'll terminate with that, but I'd like to show one slide relative to where I live and what I do in my spare time. And Fiona and I hike a lot. In Colorado, there are about 55 mountains above 14,000 feet. And I'm a long way from even close, but I've climbed a few. Last summer, Fiona and I climbed Quandary, which is south of Breckenridge, and this is the height. And we also climbed Massive, which is a little bit higher. It's the second highest peak in Colorado, and that's shown here. And this is a view from Massive. And this year, a couple weeks ago, we climbed Missouri, which is just over 14,000 feet. Fiona can scramble up these in no time. And here she was at the top of the mountain, taking a picture of me as I very carefully labored one cautious step at a time on this rather steep slope to try to get to the top. She just scrambled up it. And a lot of other places that we really like to go, like Mohawk Lakes is south of Breckenridge. It's a very beautiful hike up in the mountains. This one here, Hanging Lake, is just off the interstate in Glenwood Canyon. Take one of the rest stops, climb up a mile, and you run into this little lake up there called Hanging Lake. It's a beautiful lake, mountain stream. And this is McCulloch Gorge, which is south of Breckenridge, and this is Fiona with her llamas. And then there are lots of wildflowers that I've shown here. And then this one here was just taken at the base of Peak 9 in Breckenridge. There's a little stream that goes up to the left side there, and we found these orchids in that riverbed area growing just off the ski trail at Breckenridge of Peak 9. And then this is near Gore Creek. So I think I'll stop with this, and thank you very much, and it really is an honor to be here to help celebrate this meeting of the Corona clan and everything else associated with Gobind's wonderful career. Thank you.